Hi guys, this is Anthony from Invictus. Okay, so today we're gonna do a video on the IGCSE Biology Paper 2. Alright, and the exam is going to be tomorrow. And I hope this last minute video is gonna help you guys and do better for the exam, okay? So first of all, you have to understand the paper. For any IGCSE science paper, you will be graded on 3 criteria, okay? The first one is on knowledge and understanding. This is more recall, okay? The second one is the application. And the last one is doing some experimental skills and analysis of data given, alright? So, for your paper 2, around 50% of the marks would be on the recalling of simple biology knowledge, okay? And the remaining will be on analysis and application, alright? And that's going to make up 60 marks right here. Okay, so just follow my cursor. So let's now go through all the points for the triple science paper, okay, this paper 2, okay? And remember to like and subscribe and even comment on our video if you like it, alright? So let's look at the first one. To understand the function of enzymes, which can be affected by the changes in the active site. Okay, now, so here we have a graph, okay? So this is the rates of reaction versus the pH graph, okay? And you can see that for this enzyme, the optimum pH is around pH 7, okay? And on either side, alright, you can see that it's going to be denatured, hence the rate of reaction is going to fall. So what the rate of reaction is reflecting is whether or not there's a change to the active sites or not. And in this case, you can see on the right hand side and left hand side, because of the change in the active site, it cannot bind to the substrate, and so it cannot catalyze the reaction, alright? Now here, this is the temperature and rate of reaction graph, okay? This is from the double science syllabus, alright? But just, a small reminder that for the temperature graph, it goes up slowly upwards from lower temperatures, because with a colder temperature, the enzyme and substrate will not be colliding that fast together, okay? And then once you go beyond, let's say in this case, beyond 38 degrees, it's going to denature and have this shape, alright? So you can see that the two guys have a distinguishedly different shape right here and over here. Okay, now you have to know that it's important for plant cells to stay turgid. Okay, so let's look at the one in the center first, alright? This is flaccid, alright, and basically there's some water inside the cell vacuum, okay, but it's not like really filled with water, okay? When there's more water going into the cell, okay, let's say it goes into an area with a lot of water surrounding it, high water potential, water is going to move in. You can see the yellow arrows right here into the cell, and the cell swells up, okay? And you see the vacuum is much larger and filled with water, okay? And this is a turgid cell. This turgid cell is going to be able to help the plant maintain being upright, okay, straight, okay, which is really good, okay? On the other end, okay, you could become plasmolized when your cell is really deficient in water, okay? So this is not too difficult. Now, for the next 2 bullet points, you have to know that a balanced diet will contain good proportions of carbohydrate, protein, lipids, alright, they will provide you energy and to make the monomers to build up your other muscles and cells, okay? For vitamins, you have to know A, C, E, okay, vitamin A, C, and E. For vitamin A, it will be for the retinal cells, okay? For vitamin C, it's for cell-to-cell adhesion. For vitamin E, okay, so we have A, C, and E. Okay, is it ACE? Oh, sorry, it's ACD. Whoops. Okay, so yeah, for vitamin D, helps with the calcium absorption. Now for the minerals, you have to know about the iron, okay, and also for the calcium. Alright, so calcium is for bone, iron is for hemoglobin, and then for here, energy requirements, this will be related to how much activity you do during the day, like a lot of exercise, your age, and whether you're pregnant or not, okay? One of the examination questions were, okay, if you're like a pregnant lady, okay, why do you have more energy demand, okay? Why do you need more energy? And basically you have more energy to allow your fetus, your baby, to respire. Hence you have to intake more food, you have to respire more to provide energy for the fetus, okay? Here you see a simple setup for the calorimeter experiment, but not and it's emphasized, not for looking at the energy content of food sample. So first of all, this cell tells you that you need a thermometer. Okay, basically you will be able to measure the temperature change. Okay, and now imagine this plastic cup, okay, and replace it with a glass beaker, and beneath your beaker you can burn like a sample of food, maybe like a marshmallow, right? And you are going to measure the temperature change of the water. Now you have to know the mass of the water, you have to know the temperature change, and you have to know the specific heat capacity of water, okay, which is like 4.2. Okay, so basically your equation would be delta H equals to mc delta T, the mass of the water that you're heating up, the heat capacity of the water, and the temperature change which is caused by burning that sample of food. Alright, so this is your experiment that you need to know. Okay, now over here you see that in this diagram we have something called a respirometer. For this bullet point, you need to know you can measure the CO2 given off, okay, and any heat, and use that to reflect the amount of respiration done by an organism. Hence, we call this a respirometer. Now, on the side of A, you will see that you have a test tube right here, and inside you will have an insect, and you can also use like maybe a respiring seed. Okay, just a seed. Okay, and then inside here you have sodium hydroxide solution, which is going to absorb carbon dioxide. Now, so at the beginning on this side you have the oxygen and a small amount of CO2 and a respiring organism. And on the other side you have a control. You can fill this with something inert, something unreactive like glass beads, okay? And this is going to basically act as a control, and it could be the same mass as the insect on the left hand side. Alright, now in the center what you have here is a manometer. For this manometer, what's going to happen is that when one side have less pressure, the liquid is going to move up that side. So let's say imagine at the beginning you have it at here and the reading was maybe just like 0, and then you put in the insect, you make it airtight. After maybe 1 hour it moved up like maybe by 2 cm. Okay, so the respiration rate will be 2 cm per 30 minutes. Okay, and then just do division. Okay, and basically it's going to measure the rate of respiration, because as your insect is using up oxygen to respire, the sodium hydroxide is going to absorb the CO2 being released by the insect. Okay, hence there will be a fall of pressure on point A, and the liquid inside the manometer is going to move upwards. Okay, so this is the one over here. Now for 2.4, understand that the respiration continues during the day and night, but the net exchange of carbon dioxide and CO2 and oxygen depends on the intensity of light. Let's look at the graph over here. Okay, you see the green line represents the photosynthetic rate, and the red line represents the respiration rate. So basically, the plant will keep respiring no matter the light intensity. However, when the light intensity is high, you'll do photosynthesis, okay, and you will have a net release of oxygen. But let's say in any areas where your red line is above the green line, okay, this is when you have a net release of CO2. Even though the plant could still be doing a little photosynthesis, it's not enough to produce the oxygen required for the respiration. Okay, so basically this is the graph, and you have to understand that the amount of light determines whether the plant is giving off more oxygen or more carbon dioxide. Okay, so far. So moving down here, you have to know that you can use an indicator, and this is the hydrogen carbonate indicator. And basically when it's yellow, there's more CO2 present, and when it's red, there's less CO2. So let's look at this apparatus. Okay, you have air coming in. This air is going to contain both O2 and CO2. As it goes through this first conical flask of sodium hydroxide, which is going to absorb the CO2, okay, the oxygen that goes onto the next flask is only going to contain oxygen. And you see that the indicator is red, okay? Now, as the air keeps going over to the next flask, you have something which is respiring inside this conical flask. And what's going to happen? It's going to use up the oxygen and convert it into carbon dioxide because of respiration, alright? And then following on, the indicator will become yellow, okay? And basically you have to know the color change. It's not going to be given. You have to know how to use it, and this is how you use it. Now, the next bit is a bit wordy, okay? You have to know that inside plants, you have a vascular bundle, okay, which is composed of xylem and the phloem, okay? The xylem is actually dead tissues, okay? It's not non-living, so all it will do with the help of capillary action and transpiration at the top of the leaf, water will move out and it's going to carry dissolved minerals. Now, you have to know the definition of transpiration is the loss of water vapor from the leaf. And this transpiration stream is only one direction, okay? For the phloem, however, it's a bidirectional flow and it requires active transport to move things up and down. And it's going to be moving sucrose and amino acid. Now, sucrose is composed of glucose and fructose. And basically, you know, where you need sucrose, you can break it down, turn it into glucose, and your plant can use it for growth or, you know, respiration. Okay. Now, the next point, okay, we're going to jump to a bit of human physiology right here. So this is all about vaccination, okay, where you have the manufacture of memory cells. So how does vaccination work? Number one, you have a weakened form of an antigen, okay, or you can say a weakened form of a pathogen, which on the surface you have the antigen. We inject it into the bloodstream, okay, and what's going to happen is it's going to activate your lymphocytes. Now, as your lymphocytes are activated, it will become two types of cells: plasma cells and memory cells. Plasma cells will produce antibodies, and your memory cells will be retained inside your bloodstream. And if one day you encounter the same pathogen again with the same antigen on the surface, you're going to have a much faster and more greater response, okay, of antibodies released, so you will not get sick for that long. Alright, so this is the principle of vaccination, and the reason why you have a better response in the future is because of the memory cells right here. Okay, so we have a small diagram, okay, the plasma cells making the antibodies, which are made of protein, okay, it's going to bind to the pathogen, and this pathogen could be, you know, broken down by the antibodies, or maybe the antibodies will helps the phagocyte to engulf this pathogen matter. Okay, the next bit. Now, platelets are essential to blood clotting. So what's going to happen is that when you, let's say I get injured over here, okay, and start bleeding, the platelets nearby are going to start producing clotting factors. Now the clotting factors are going to convert prothrombin to thrombin, okay, and basically this prothrombin conversion, okay, the thrombin becomes an activated form of the enzyme. It's going to convert fibrinogen, which is a soluble protein, into fibrin, which is an insoluble protein. Now this insoluble protein, imagine it's like a strand, and this strand is going to start cross-linking to form a network, and this network is going to trap blood cells and then forms a clot. Okay, so going down. So now we're looking at the eye. Now a lot of students are confused with the structure of the eye, so let's just go through briefly. Okay, follow my cursor. Alright, over here this is the cornea, okay, and then over here there will be the iris and the pupil. Alright, in our next slide you're gonna see a better diagram of the iris and pupil. Okay, the pupil lets the light in, okay, and then here we have the lens, and attached to it would be the suspensory ligaments and also the ciliary muscles. Okay, Now, for the first one, for this diagram, what's going to happen? You're looking at an object which is quite far away, okay? So your lens have to be stretched thinner. Now, on the surrounding lens, you're going to have the ciliary muscles relaxing. When the ciliary muscles relax, your suspensory ligaments are going to be more taut. It's going to be tighter and pulls the lens like that, thinner shape. But now, on this side, if you're looking at an object which is much closer, okay, you will have a contracting of the ciliary muscles, and your suspensory ligaments will be less tight, and the lens will be thicker, and it's able to focus the light onto the back of the eye at the retinal layer. Now, somewhere along here, you're going to have an optic nerve. Okay, so go back to your double science knowledge if you're unsure about it. Now, let's move on. Here we have a picture of an eye, and you see the pupil is quite large. So this is the environment when there's not too much light. Okay, you can see that the pupil is dilated, and the iris is like enlarged. So what's going to happen is that you have to know that in this scenario, the radial muscles of the iris contract, okay, and the circular muscles relax, and then this enlarges the size of the pupil, and so more light can enter. Now, over here, in this diagram, you have a smaller pupil, and you have a contraction of the circular muscles. Okay, it becomes smaller. Okay, so less light will go into my eye. Oh, so when the environment is really bright, this is what's going to happen. Moving on. Okay, so on this slide, you will see that you have to know how our skin is going to help us with temperature regulation. Okay, so basically what's going to happen? When let's say I'm really hot, what's going to happen is that my blood vessels in my skin is going to dilate. We call this vasodilation. When this dilates, what's going to happen? The blood will be closer to my surface of the skin, and it's easier for heat loss. At the same time, we're going to start sweating, and the sweat is going to evaporate from my skin and take away the heat. Okay. Now, what happens if it's really cold? The opposite happens. There's no sweating. Instead, you are going to have vasoconstriction. This is why some people will appear more white. And then, at the same time, you're going to shiver. And when you shiver, it's actually small, rapid muscle contractions, which are going to produce more heat. Is that okay? Now, EC3 marker. So, conditions for seed germination. So, water, oxygen, and a suitable temperature. Okay, water to rehydrate the seed tissues, oxygen for aerobic respiration, because anaerobic respiration is not suitable, okay, because it could produce ethanol, and the ethanol is going to reach toxic levels inside the seed. Okay, now at the same time, for a suitable temperature, because you have enzymes to help turn your seed, germinate it, and form a new plant. So over here, we have the seed. It starts by forming really baby roots, okay, which start absorbing water and more minerals, and then it forms a baby shoot, okay, and then this is going to take over to photosynthesize, and you don't have to use the food reserve inside the seed anymore. But at the very beginning, the type of food reserve you have inside the seed is actually starch, and starch can be broken down into maltose and then into glucose. Alright, so moving on. So here we have the placenta, alright, and also the amniotic sac. So what's going to happen here? For the exams, you have to know that the amniotic fluid is going to protect the baby against like shock, any impact. Okay. At the same time, the placenta is in charge of the exchange of nutrients between blood from the fetus and also blood from the mother. All right, the blood will not mix, so there will be in close proximity, and there will be the exchange of nutrients, maybe let's say glucose and oxygen, and the rates could be urea and CO2. Okay, and this is what's going to happen in here. Then moving on. Now here let's do a bit of revision about genetics. Okay, several terms: dominant, recessive, homozygous, heterozygous. The triple science point is actually focused on the codominance right here. Okay, so I'm going to use blood groups as my example. Now, for dominant alleles, it is going to be expressed regardless of what is it paired with. Okay, so that's dominant, it's always expressed. Recessive, it will only be expressed when you have two copies of the recessive alleles. Okay, so we call that homozygous recessive. Hence, homozygous recessive means two recessive alleles, and homozygous dominant is two dominant alleles. Okay, for heterozygous, I do not have to specify whether it is dominant or recessive, because it must be paired with two different alleles, and that would be the meaning of heterozygous. Now, the phenotype means the physical characteristics expressed. Okay, so what you actually see, okay, the color of my hair. Okay, now for the genotype is the combination of alleles in the organism. Okay, so now let's look at the blood group example to work out the codominance. Now, for the blood group O, you have two recessive alleles. For blood group A, you could be homozygous dominant A or heterozygous A. And for B, heterozygous B or homozygous dominant B. Okay, but now for blood group AB, you're gonna have a heterozygous, but this time it's both the dominant allele A and also dominant allele B, and together phenotype AB. Okay, and the next bullet point, this is more focusing on the mutations. So basically, if you're exposed to ionizing radiation, all right, or if you're exposed to some chemical mutagens, okay, it will increase your risk of having mutation, and mutation of cells could cause cancer, and basically your tumor cells could have uncontrollable mitosis. So this is what happens. So stay away from ionizing radiation and any chemical mutagens. Now, the next bit, we are now on topic 4. We're going to focus on the water cycle, a quick review of the carbon cycle, and the most difficult nitrogen cycle. Alright, so right here we have described the stages in the water cycle, so including evaporation, transpiration, condensation, and precipitation, which means rain. So on this side of the diagram, we have evaporation of water vapor. Okay, it's going to condense into clouds, and from the cloud it's going to rain precipitation back, and then on the layer of the mountains it could have a surface runoff, and the water will soak in the soil to become the underwater and underground water supply, and it could be in lakes, it could be in rivers, and go back to the ocean, and the whole cycle starts again. In the center of this, you could have transpiration from plants to release water vapor back out to the clouds. Okay, so the next bit is all about the carbon cycle. So imagine we have carbon dioxide or carbon in the air. Okay, so the only way it's going to come back down is by photosynthesis. At the same time, it could be by the uptake by oceans as well. Okay, but that's not too focused on in the IGCSEs. Okay, more in the IB and A levels. Okay, so photosynthesis, CO2 goes into the plants to form organic compounds. This plant is going to respire, so hence release CO2, and then animals can eat the plant, so also respire and release CO2. Imagine many years ago, decades ago, you're gonna have the plant and the animals to form fossil fuels. We can combust fossil fuels to release energy to make it into electrical energy, right? And it's going to release carbon, okay, in the form of CO2 again, and this whole cycle just carries on. Okay, moving on. Okay, now this is the more difficult cycle, I would say. Okay, so for most students. Okay, let's begin with the nitrogen in the air. Okay, the diagram is a bit small. Okay, just follow my cursor. This is nitrogen N2 in the air, and there's several ways it can come down. Now, with lightning energy, it can also turn it into a nitrate form. Okay, but more commonly we have nitrogen fixing bacteria right here. Okay, we're going to turn it into ammonia compound, sometimes ammonium ions. Now, these ammonium ions in the center of the nitrogen cycle can now become, with the help of nitrifying bacteria, become nitrite and then become nitrate. This nitrate is the form where plants can uptake into the roots with the use of active transport and turn it into proteins. Okay, or amino acid and then proteins. Okay, now, but if you have nitrifying bacteria, you're also going to have denitrifying bacteria. Now, for the nitrifying bacteria, it turns the nitrate back into nitrogen. Okay, and then now, with the help of decomposers, it can turn proteins inside animals and plants, and then turn it back into ammonia as well. So that completes the whole cycle. All right. So go to on Google and search up, nitrogen cycle and think about it more. It could come up. Okay. Now, the next bit is about eutrophication. So let's say you use a lot of fertilizers. Okay. Look at point number 1. Okay, which contains nitrates, potassium, and stuff like that, and basic and phosphates, and then it actually leaches into the nearby lake. Okay, so any stagnant water. Okay, what's going to happen is that these compounds from fertilizers are going to provide the nutrients required for, let's say, the algae, and you get an algal bloom. Okay, massive growth of algae. What's going to happen? This algae is going to block off the sunlight, so it's not going to reach the plants underwater, and plants are going to die. And then the bacteria inside is going to decompose any dead tissues and use up all the oxygen. Okay, and using up the oxygen, we call this the oxygen burst. What's going to happen? Fishes, aquatic life is going to die inside the lake, and this is all because of the leaching of fertilizers into the lake. Okay, moving on. Now, this is focused on the production of yogurt, and you have to know about a bacteria, and this bacteria is lactobacillus. Okay, so take a look at the diagram first. All right. So, how do we make yogurt? Now, number 1, we have milk. Okay, it's gonna mix, and we do something called pasteurization. And basically, it's gonna heat to a high temperature and then cool back down. It's gonna remove the oxygen and kill harmful bacteria. Then we cool it down to a temperature around 40 degrees. Okay, because this is the optimum temperature for lactobacillus. Okay. Now, what's going to happen is that yogurt bacteria, which is added inside, is going to ferment the lactose, the milk sugar inside the milk, into lactic acid, hence the acidic taste when you eat yogurt. All right, and then you let it do the fermentation, and then you store it. Okay, and then it becomes yogurt, and you can eat it. All right, so you have to know that for the lactobacillus, it is for the fermentation. Okay, and turn lactose into lactic acid with anaerobic respiration. Now, for a fermenter, okay, you have to control the conditions inside. The easy part would be, to control the temperature, to control pH. But you have to be able to interpret and label this diagram. Okay, a fermenter. So what's going to have it? An agitation system where it's going to just mix the nutrients and the enzymes together, and make sure that the heat is well distributed inside the whole solution. Okay, you're going to have some sensors which is connected to a system to monitor the conditions, pH or temperature, okay, and then basically you could add oxygen inside, you see an air inlet there, okay, if you're feeding pump to put in the medium, and then the air, okay, to sometimes if you want aerobic respiration, then you put in more air inside. If you want anaerobic, then you just close the air valve right there, okay, and then you can get your products. All right, so you have to know what the functions of each component, okay, and what's the function of fermenter overall. Okay, we're near the end. Now, the last bit is about transgenic organisms. It seems like an impressive term, but actually it's really simple. Transgenic is when you take genetic information from an organism and then you give it to another organism. So from one species to another species, that would be called transgenic. In this diagram, all we're taking is in the blue is a beneficial gene, the gene that we want, and then maybe we use a plasmid, all right, and we add it to a plasmid and form a recombinant DNA, and this recombinant DNA could be absorbed by bacteria, and the bacteria is going to have this gene inside. For this situation, you have to think back to your genetic modification, okay, how to add genes using restriction enzymes, ligase enzymes, all right, recombinant DNA, maybe taking the insulin gene from humans and then making it into a recombinant organism. Put it in the fermenter, so you'll start producing insulin for us, for diabetics, okay? Now, for the last point, now, for some reason a lot of schools will usually miss this out, okay? So here, evaluate the potential for using cloned transgenic animals or cells, okay, to produce human antibodies or organs for transplantation. So how would this work? Okay, so you will inject a pathogen into, let's say, mice or even dogs, okay? What's going to happen is that the white blood cells, the lymphocytes inside, is going to be activated by that pathogen. So think back to our vaccination, okay? It's going to produce the lymphocytes that can produce the antibodies to tackle this pathogen, right? Now, I can take this lymphocyte or the plasma cell and I merge it with a tumor cell, okay? On merging of that, it can form cells called hybridomal cells. Now, I put this in bracket because this is not required. Knowing this hybridomal term is not required for the IGCSEs, okay? So, this hybridomal cells is capable of continual mitosis, at the same time keep producing antibodies because it's merged with a lymphocyte, okay? You put in the fermenter again, provide the optimum conditions, and then you produce a large quantity of antibodies. And what you have to do at the very end, just purify the sample, okay? We have these antibodies and you can use it to treat diseases, okay? So now, this is the end of the video. Please subscribe, and I hope you enjoyed the video, and comment, okay? If you guys really like this video, just let me know, and I'll make it for maybe for other syllabus as well, maybe for the triple science chemistry, which is upcoming next week, or the physics paper as well, okay? So I hope you guys like it, I hope it helps, okay? And if you need any tutoring, the details are inside the description down below, so check it out, alright? And see you guys soon. Bye.